I will be making some forward-looking statements. So at ORA, we're working to transform the treatment of patients with choroidal melanoma. We have developed a very novel targeted therapy with a unique mechanism of action, and we received orphan drug designation in 2015. AU-011, our flagship drug, will be in the clinic next year and will have the potential for early clinical readout. And if the drug proves as we believe it will, we have a great opportunity for an accelerated regulatory path forward. In addition, because we're developing a novel class of drugs, our platform can have very nice applications in non-ocular cancers as well. And we've built a management team that can do this very efficiently with the expertise in orphan drug development and commercial launch. So let me tell you a little bit about choroidal melanoma. It is a rare disease, and currently there are no FDA-approved therapies. Patients have been treated the same way for 50 years. The opportunity is that this disease is diagnosed really early in the primary setting. And these patients are seen by retina specialists, a subset of ocular oncologists. This is an extremely focused call point. There are about 54 ocular oncologists in the US and 52 in Europe. Patients diagnosed early are non-metastatic, and about 25% of those will metastasize in 5 years. There is no approved therapy either for the metastatic disease, and it's a life-threatening disease. So the opportunity to diagnose a cancer so early, not yet metastatic, and have nothing for these patients is remarkable, and that's what we will try to improve. So as I mentioned, for half a century, this is how we treat patients with choroidal melanoma. These are a combination of radioactive and surgical procedures, basically placing a plaque filled up with iodine radioactive seeds in the back of the eye. As I mentioned, it requires multiple surgeries, causes severe vision loss, over 6 lines of vision, and does not prevent metastasis. If the melanoma is too large, the only available treatment option is enucleation. So why are we excited at ORA? For the first time, we have something to offer to these patients, and we believe is unique and incredibly powerful. So we've developed a novel class of drugs, what we call viral nanoparticle conjugates. They are based on the capsid of a virus. They do not have any genetic material. They do not replicate. But this capsid is conjugated to a small molecule. That's what provides the anti-cancer activity. So the viral nanoparticle is administered in the eye intravitreally, and it binds the tumor really efficiently. These tumors overexpress heparan sulfate proteoglycans that are highly modified, and the virus is capable of binding to these with high affinity and avidity. Upon light activation with an ophthalmic laser, the small molecule is capable of liberating free oxygen radicals and creates an incredibly powerful necrosis. This is a mechanism that is highly reproducible. It doesn't depend on the molecular pathway of uveal melanoma, which is highly complex. But as long as the drug is there, this necrosis will happen. So how will we develop this, and how is it administered? So the beauty of this, if you remember the very complex surgeries and radioactive treatments, this is an efficient outpatient treatment protocol. First of all, it's an intravitreal injection. Luckily, after 10 years, we think intravitreal injections are standard of care in ophthalmology, same as Avastin, Eylea. It is activated with an ophthalmic laser 6 hours after the intravitreal injection. And this ophthalmic laser has already been used for years. It was introduced with the combination of Visidyne, and it uses 689 nanometer laser light to activate the drug. This is exactly the same wavelength which has demonstrated to be safe for the retina for Visidyne. So what data do we have to date? We have developed and worked with the best ocular pathologists to develop a model for choroidal melanoma. This is a rabbit model where we inject uveal melanoma human cell lines in the choroid of the rabbit, and it grows exactly the same as the human disease. The rabbit model also allows us to do an intravitreal injection and laser activation, so this highly resembles a clinical trial. With this model, which has been replicated in 3 different academic labs, with 2 injections of the drug once every week, plus laser activation, we have complete responses, in particularly those tumors that are small. This data is remarkable. It's just 2 or maximum 3 injections to eliminate a tumor that is highly aggressive. So let me show you some of the histopathology, because the importance, obviously, is to see that this says or does what we think it will do in the clinic. So what you see here are the histopathology of 2 animals, one control and the other that has been treated. And what you can see is the growth of the melanoma in the choroid. This model is highly aggressive, so if left untreated, it grows outside of the eye, extraocular extensions. In the treated animal, what we can see is that there's no tumor remaining and there's no damage to the overlaying retina. So where are we in our development? Next year is an incredibly exciting moment for our company. And we have very short-term, as I mentioned, clinical readout. First, we will demonstrate safety, but we have the opportunity to also demonstrate efficacy. And as I mentioned before, once we show efficacy with this drug, it's highly likely that it will reproduce. And as there is no other alternative for these patients, we do think that we will have an accelerated path forward for registration.